Hello, my name is Akeel Faizal, and I will be presenting to you my research on the in vitro evaluation of lytic bacteriophages against Shiga toxin producing E. coli 0157H7 on human intestinal cells. So Shiga toxin producing E. coli 0157H7 is one of the most common serotypes of STEC involved in foodborne illnesses and causes diseases such as hemolytic uremic syndrome and hemorrhagic colitis. Although antibiotics are effective against many bacterial infections, studies have shown that it is not advisable to use antibiotics against STEC due to the augmented release of Shiga toxin from dying bacteria. These Shiga toxins released are what lead to hemorrhagic colitis as well as hemolytic uremic syndrome in humans. So in the absence of reliable treatment and control measures against STEC infection, phage therapy has been increasingly investigated for the control of STEC infection, and for the use in biocontrol, we are interested in lytic phages, which will lead to the lysis of bacteria. So moving on to our cell culture model, enterohemorrhagic STEC colonizes human and animal intestinal epithelial cells, and when choosing an in vitro cell culture model to use, we had chosen two human intestinal cell lines, CACO2 and T84 cells. The identified problem for the research was that the effectiveness of bacteriophages in preventing STEC 0157H7 on intestinal epithelial cells is unknown. And so my hypothesis was that STEC 0157H7 is able to attach to the human enteric epithelial cells, and this process can be prevented via bacteriophage treatment. So moving on to the materials and methods, my first experiment we'll look at is a qualitative attachment assay for STEC onto the CACO2 and T84 cells. The qualitative assay was done using 24-well plates, then gelatin-coated cover slips were placed into the 4 wells. CACO2 and T84 cells were then seeded into them, and then the wells were infected with EDL933. After 1 hour of incubation in 37 degrees Celsius and 5% CO2, the cells were washed and stained with Game Star stain in order to visualize bacterial attachment. The cover slips were then placed on slides and read under a bright field microscope. From the qualitative attachment assay, we were able to visualize the attachment of EDL933 onto the CACO2 and T84 cells. You can see the circular nuclei of the cells, which are shown by the red arrow, and STEC with the green arrow. So next experiment is our phage treatment of STEC infected intestinal cells. And so the prophylactic phage treatment of human intestinal epithelial cells was done in order to prevent STEC from attaching onto the cells. First, 24-well plates were seeded with CACO2 or T84 cells, and then 1 hour before infecting with bacteria, the phages were added into the wells. Then, in order to achieve an MOI of 1, STEC suspended in attachment media was added into the wells. And then we have our various controls. So the first phage we looked at was AKFV33, and the y-axis represents STEC count in CFU per ml, and for the x-axis bars, the blue bars represent the attached infected CACO2 or T84 cells with phage versus without phage. The red bars represent the amount of bacteria found unattached with phage versus without phage, while the gray bar shows the amount of bacteria in cell-free media with phage versus without phage, and we had observed that AKFV33 was able to decrease STEC attachment in both the CACO2 and T84 cells by about 3 logs. Then in the unattached supernatant for both cell lines, there was a 3 log reduction, while in the phage and STEC containing no cells, we saw a 4 log reduction, and the group differences were seen as highly significant. Then we looked at cells treated with a phage cocktail of 3, which contained the phages AHP24, AHS24, and WV7. In the CACO2 cells, we saw a 3 log reduction in the attached bacteria, whereas in the T84 cells, we only saw a 2 log reduction. In the unattached bacteria, we saw a 4 log reduction in the CACO2 cells versus a 3 log difference in T84 cells, while in the cell-free media, we see a 4 to 6 log reduction in bacteria in both. So despite the reduced differences in attached and unattached bacteria on the CACO2 and T84 cells, the data was still considered highly significant in both. Another interesting thing to note was that of the bacteria treated with phages that was counted, we did see some muted STEC as noted by changes in the colony morphology when compared to the regular colony morphology of EDL933. To conclude, we were able to establish a proper visual representation of STEC attachment onto the CACO2 and T84 cells, and then for our phage treatment, we saw AKFV33 had a better phage lytic abilities against STEC EDL933 attachment on the CACO2 and T84 cells than phage cocktail 3. Future experiments include forming a virulence assay and whole genome sequencing to determine if the mutants are truly resistant or contaminants, as well experiments for testing safety of our phage treatment by looking at Shiga toxin levels in phage treated with STEC versus phage free of STEC using ELISA. The results of the probe's work will contribute to the knowledge of therapeutic outcomes of phages against STEC 0157H7. I would just like to end by thanking my supervisor, Dr. New, for all her help and mentorship throughout the summer, as well as Makila for providing us with cells, and everyone in ULab and the virology group for their support in my experiments.